goes into it and out of it. So that is going to be the lab that we are going to be working on today, is we're going to be looking at how things move into the cell and out of the cell by creating artificial cells, and also looking at plant cells underneath the microscope. Okay, so remember that organelles are membrane-bound structures that are found in the eukaryotic cell. And remember that we have different ones of these, and they have different functions depending upon what they are. So one function of the rough ER is to produce proteins. And so this is a diagram from your book. And it produces proteins on the ribosomes, and then they get put into a vesicle. And the vesicle then gets transported to the Golgi apparatus, which is sometimes likened to a sack of pancakes, where it gets modified. So the protein becomes modified, then it goes back into a vesicle, and then it is secreted to the outside. And so we have the production part of the cell, the assembly line, and then we have packaging, plant, and modification, and then we put it to the outside of the cell. Okay. So let's look at a very specific, specialized type of cell that is found in mammals, found within mammary glands. And mammary glands are interesting because they are modified sweat glands that are used to transport nutrients to the newborn. So we have milk glands in the breast tissue that produce a very complex substance that we refer to as milk. So this handout that I'm giving you has a description of what is typically found in breast milk. This would be humans, but they could also talk about the breast milk of other mammals. And then what is typically found in formula. So formula would be what you would buy at the grocery store in order to feed the baby instead of or in addition to breast milk. Okay? So if we look at this, this is a complex substance that contains lots of interesting things that are produced by very specialized cells. So we have mammary gland cells. And they secrete milk. When we look at the components of milk, we find that there are lipids. Specifically triglycerides, which would be like animal fats, and that tends to be saturated, right? So it'd be like a saturated triglycerides. So this is animal fat. So when milk appears, actually that's white. It also appears white when you remove the fat, but it appears fatty, right? It has the triglycerides. Whole milk, the fat will float to the surface, right? And then you can skim the fat off the top in order to remove it, and then you have the other milk buildup. So remember that the lipids are not soluble in water, but they're secreted. So where would the saturated triglycerides that are found in the milk, where in that specialized mammary gland cell would they be produced? Do you remember from what we just talked about, the different organelles? So we'll put components of milk, and then I'll put their production over here. It's in the endoplasmic reticulum, but not the rough one. What is the other one called? Smooth. Okay, so this occurs in the smooth ER. Okay, the smooth endoplasmic reticulum. So if you look on the next page, okay, or if you flip that page over, okay, you see that there is some smooth ER. So, yeah. So the smooth ER would be like right here. It says that lipids are synthesized here. And remember that lipids just simply can pass through the membrane. So we talked about how lipids can fuse with the membrane and then pass through. So this lipid just kind of floats. We're going to talk about the cytoskeleton that helps to transport stuff. But the cytoskeleton helps to transport it to the outside, and then it is simply secreted into the milk. There's also what they refer to as long chain fatty acids. So this is another type of saturated triglyceride. This is saturated, so it's a long chain fatty acid. And this is actually believed to be really important for the development of your nervous system and the baby's nervous system. And so that would also be produced by the smooth ER. Recently, they've started adding it to milk. So specifically in organic milk, there's regular organic milk, and then there's this organic milk that costs like $1 more per half or quarter, whatever you buy, half gallon. What is that called? Does anybody know? What do they add? What is added to milk? It's abbreviated. It's 3 letters. DHA, right? So this is DHA. Okay, so this is nervous system development. So I'm sure that you could get formula that now has DHA added. But in general, if you look at this, if you go down to fat, it says long chain fatty acids. It says cow's milk formula doesn't have any, right? So that is something that's found in breast milk that typically isn't found in formula, but might be important to have. So that's why they put it in there. And if you have children, you might want to pay the extra money so that they're getting that. But they can also get it elsewhere in their diet. Okay, so that would be the lipids. Also notice that they also have, forgot to mention, they also have cholesterol. So what is cholesterol converted into that is important in our body? Cholesterol is a steroid, and it's a precursor to vitamin D, but it's also a precursor to our steroids, which would be what? Estrogen and testosterone. So cholesterol is a precursor to the other steroids and vitamin D. It's also really important to have cholesterol in our diet because it, we'll talk about this, but it is also found in our plasma membrane of the cell. Okay? So typically, cow's milk formula does not have cholesterol. Okay, so let's move down. Those are the lipids. We also have proteins. So if you take milk and you add vinegar, what's going to happen to that milk? It's going to not necessarily spoil, but it will, what's it called? Curdle, right? And so that curdling is the proteins becoming denatured, and they change shape, and they come out of solution. Now, curdling could be due to having bacteria or fungus get inside the milk and actually produce acid, which makes the milk go bad, right? So that's when you know your milk is bad. So if you lower the pH, the protein that comes out is the casein. So casein is a primary protein that is found in milk. And so it would be like the curd, right? So it would be the curds. So sometimes you can go to cheese factories and get cheese curds, right? That's protein. It generally doesn't have any fat in it. There are other proteins that are also important, which include antibodies. And these have an immune function. So the first milk that is produced by a lactating female has a special name. Does anybody know what that is called? The first milk is called colostrum. So colostrum is the first milk. And if you've ever seen the first milk that's comes out, it tends to not look white at all. It tends to be really thick and it's kind of yellowish, right? And that first milk is important because it provides passive immunity to the offspring. So they get antibodies, right? And then those antibodies get transferred from the mother to the baby. And so it's passive immunity for the offspring. So the mother is exposed to pathogens like microorganisms and viruses in the environment, and she'll have antibodies circulating in her blood, and some of those antibodies then pass across the placenta, and then when the baby's born, instead of across the placenta, they get it in breast milk. Right? So you'll notice that if you look at your sheet there, right, there is no antibodies that are found in, so no immunoprotective. So those would be antibodies. So the immunoprotective factors, like IgA would be one, would be an example of an antibody. So that's why they recommend that women breastfeed for at least like 6 months, because it helps keep the baby from getting sick. And so if you just give formula, then the baby won't get that passive immunity. So where is the proteins that are going to be secreted? Where are they produced in the cell? We just had this on the quiz. Rough-ER. Now you notice that there's another one I forgot to write down here, enzymes. So enzymes are found in breast milk, and they help the baby digest the milk. So the enzymes get ingested by the newborn via the milk. It helps to break that down. Okay. We also have sugars in the milk. And does anybody know what sugar is found in milk? What is the milk sugar? Lactose, excellent. Okay, so that's milk sugar. That would be a source of energy for the newborn, right? And they are able to digest lactose and turn it into glucose and galactose. So because lactose is composed of glucose and galactose, what kind of carbohydrate is it? What kind of sugar is it? Disaccharide, right? 2 components. So lactose is like sucrose, like we looked at last week in lab. The lactose is a disaccharide that instead of glucose and fructose, it's glucose and galactose. So there's a specific enzyme that is used to digest lactose. And the enzyme is called lactase. So remember I said that anytime you see that ase ending, that means that it is an enzyme. So lactase digests lactose. This is produced in the intestines of the newborn, and most newborns are able to digest it, digest the lactose. But most mammals lose the ability to digest lactose upon weaning. Which is why you're not supposed to give your cat and dog, adult cat and dog milk, right? Most mammals lose ability to digest lactose upon weaning. Right. The reason why is that you don't want to give it to them is instead of them digesting it, it gets digested by bacteria in the large intestine, and it produces gas, which causes the intestine to bloat, right? It can also lead to diarrhea, which was the loss of water in the feces, which is a really bad thing because that can lead to dehydration, right? And it could just cause overall pain, right? So lactose intolerance. You said here, like, I am lactose intolerant. You hear some people saying that. That means that they can't digest lactose, but bacteria do. And this causes gas. Okay. So it can be very painful. This is not an allergic reaction. So an allergy to milk would cause inflammation, it would cause tissues to swell. So this is just that the bacteria that are present in our digestive tract will eat the lactose and then they'll grow and they'll develop and they'll produce more bacteria and it just causes bloating, right? And pain and intestinal issues. Okay. So in lab today, we're also going to watch a video that talks about why we see in certain populations of humans that we now have the ability to digest lactose, even as adults. But you can make milk more digestible by doing certain things to it. So if I wanted to make milk more digestible, what might I do to it to make it more digestible for everybody? You take the lactose out of it, right? So you can see that we can get lactose free. Right? But what have we done a long time, for centuries? What have we done to our milk? Well, we heat it up, but that actually kills bacteria. But we actually do what to our milk to produce other good things that we love? Cheese, big cheese. How does cheese come about? We add microorganisms, right? So if I take my milk and I add certain strains of bacteria, I can get Swiss cheese. And Swiss cheese is the one with the holes in it. It's got a really particular taste. And the holes are due to the bacteria producing gas bubbles. And they produce the gas bubbles, and then when it solidifies, I have holy cheese, right? And then some cheeses, you add a fungi to it. Some cheeses, you add bacteria to it. But what you do is you let the microorganisms digest it for you. And so some people that are not able to drink milk, for example, can eat yogurt because yogurt is fermented milk. So you've added microorganisms and the microorganisms have broken down the good things in it, and then you're digesting the microorganisms so you can get lots of nutrients from it that way. Okay, so we have fermented our milk to make it more digestible, which is actually the exact opposite of pasteurization, because pasteurization is heating it up to kill everything, right? To kill all the microorganisms that might have gotten in there, but also to denature the enzymes and to kill the living cells that might be in there. So does anybody remember where are sugars produced in our eukaryotic cell? Golgi apparatus. Oops, the Golgi apparatus that ran off that's US apparatus. Okay. So if we look at that, if we turn our sheet over again, okay, here we see that sugars are being synthesized in the Golgi apparatus, and they produce them, they put them in little vesicles, and then they get transported to the outside, and so this would be like lactose being secreted by the mammary gland cell. Now the proteins, we're going to talk a lot about protein synthesis and molecular genetics, but the proteins are produced on the roughy R according to the information that is found in the nucleic acid. So the DNA is in the nucleus, but messenger RNA comes out of the nucleus, attaches to the ribosomes, and then this would be protein. So the protein is produced, and then it's sent to the Golgi apparatus to be modified. And then the mitochondria would be the producing ATP, all of the energy it takes to catalyze these chemical reactions in the rest of the cell. So that would be where the energy is produced. Okay, so if you go to your table again, you can notice that there are, if we just go through some of them, breast milk contains live human cells. There would be no live components in cow's milk. Contains human hormones and growth factors, and there's none in that. No enzymes, factors that help the gut mature, none of those. No antibodies, no immunosuppressive factors, okay? And then you can look at the proteins. A lot of the proteins get destroyed because the milk is pasteurized before it is then turned into formula. We talked about the DHA and the fat. And then you get down here to miscellaneous. So why is iron really important? What molecule do we make that has iron at its center? Blood. So hemoglobin. So hemoglobin, iron is necessary for hemoglobin production, right? So iron is used for hemoglobin production. So there's more iron in breast milk than there is in cow's milk. Hemo, it's H-E-M-O, sorry, hemoglobin. And that is what transports oxygen. That is what is found in red blood cells, and it transports oxygen out through the entire body and then transports it to the mitochondria so that they can produce energy via cellular respiration. Okay, and then we see that we have some important electrolytes like sodium chloride. K means potassium. Calcium is Ca, and then phosphorus. Okay, and then we get to the very bottom, and this is probably the most controversial issue here in terms of breast milk and formula. And at the very bottom, we have what are referred to as persistent organic pollutants. Sometimes this is abbreviated. You'll see it abbreviated in the media as POPs, persistent organic pollutants. So these are organic molecules, meaning what are they made of primarily? Carbon, right? They're persistent, which means that they're lipids. So they are fat soluble. Because they're fat soluble, you cannot get rid of them via the urine. Most lipids are broken down by the liver, but if the liver doesn't recognize it because it's synthetic, then the liver does not know how to break it down. So these are also synthetic. Which means that they're not naturally found in the natural world. Okay? So if we look at some of these, you might be familiar with DDT, right? This is an insecticide, and it was used to kill mosquitoes. So you can actually see videos of insecticides being sprayed over families playing at the beach. Right? We now know that DDT will accumulate in your body, and you can't get rid of it. Once it gets in your body, you generally don't get rid of it. So it stays inside your fat and gets stored there. Okay. PCBs, these are byproducts of manufacturing. And so if you hear about the pollutants in the Columbia River, the Columbia River has a problem in this that the water moves pretty slow and they have a huge sediment layer, right, down in the bottom of the Columbia River. And manufacturing plants have been putting chemicals, including PCBs, into the river for a long time, and they settle on the bottom, and then they have to dredge the Columbia River in order to allow the big boats to come up. And when they dredge it, it gets the PCBs back in the water, and then this gets taken up by fish. So sometimes you hear about salmon, for example, that have high PCB levels in them. And it's also recommended that when you eat salmon, you're eating the PCBs, right? And that will then accumulate in your body because you can't get rid of it. And so they recommend that you limit the amount of salmon that you eat, and you be very careful from where it comes from, because it could be high in PCBs. That's very controversial to the tribes in this area, because obviously salmon is, one of their first foods. And it's very important for them to be able to eat the salmon that's caught. And so it's a very big issue for them because it accumulates in their fats. We also have another example, which is PBDE. Okay. These, and I'm not going to test you over these, but it might be nice for you to be able to give me an example of a pollutant. This is actually found in flame retardants. So if you think about those pajamas that they used to make that were synthetic, they might put a flame retardant in the pajamas. They would also, carpets tend to have flame retardants in them. And then also foam furniture. So if you have foam furniture that is like before 1990 and it starts to break down, your cat, I had this when the cat loved to scratch, right? I had pieces of foam everywhere. Well, I had to get rid of it after I did a little bit of research, because if it's old foam furniture and the foam is starting to break apart, then you can get that into your body, and then it will start to accumulate in your fat. So the reason why this relates to breast milk is that these accumulate in breast tissue. So in breast tissue and other areas of fat. So if you're a male, it'd be in your fat, right? But, in breast tissue, and ends up in breast milk. So in other countries, I don't believe we have it here in this country. I haven't looked this year, but last year we didn't. You could take your breast milk, take a sample of it, and you could send it off to a company, and they would see how much of this. We know that there's going to be some of these organic pollutants in breast milk. We just don't know how much. So in Europe, they're way ahead of breast milk monitoring. And when they started to find high levels of flame retardant in the female's breast and in the milk, they then banned this. They banned it before we did. And what they see is that once you ban them, then the levels of these organic pollutants will decrease in the body, but they'll also decrease in the breast milk. So if we look at an image that shows this. Okay. So here we got really concerned about fires, and is this United States? This is Sweden. So we got really concerned about fires, and we started putting flame retardants in everything, right? And then you see that there's this peak of, in Sweden, PBDEs, that's flame retardant. And then they subsequently banned them, and so now it's actually starting to come back down. Okay? We see DDT was really, really used back in 1970, and we start to see a decrease in it. So the controversy is about cleaning up the environment so that we can clean up the breast milk. And there's obviously benefits to breastfeeding. If you look at the table, you'll notice that there is a detriment to breastfeeding in that processing of cow's milk should reduce or exclude most contaminants. So there's not going to be these organic contaminants at the levels that they are in human breast milk. But the benefits of breast milk are believed to be outweigh this. And so the problem is that females are, as they lactate, they're downloading the pollutants to their firstborn. And it's kind of interesting, after they have had one baby, the second baby pretty much gets clean milk. So if you're a firstborn and you were the first one breastfed, you kind of get all the toxins, right? And then your younger siblings got fewer toxins than you. Okay. The other thing is that we know that breastfeeding reduces the risk of cancer in the mother. And so it could be that this accumulation of these organic pollutants in the breast are actually leading to breast cancer. And so that if you lactate, you can actually get rid of these pollutants and you'll have a decreased likelihood of breast cancer. So again, I can see a future where maybe you'll be given hormones that cause you to lactate. You'll lactate, you'll get rid of all your toxins, right? And then you'll stop lactating, and then you'll have your firstborn. And then, then the milk will be clean. Or hopefully we'll see an era where we realize that these pollutants are potentially harmful for us as a species, and then we're going to decrease the use of them. So one problem with these hormones, specifically DDT, is it is an estrogen mimic. Okay? So it acts like estrogen does. And one of the things that estrogen does is it causes cells to divide. And so it could possibly, as an estrogen mimic, could possibly cause breast cancer. Okay, so the takeaway is that our breast milk is polluted, just like our environment is polluted, but we have lots of benefits. And so there's lots of resources out there if you're interested to go online, for example, and find out ways that you can reduce your risk of being exposed to these pollutants. Any questions about that? You could also do your extra credit on possibly this topic. This is a good topic. Okay. So I want to talk about a little bit about the other structures that are in the cell that I haven't talked about yet. And one of these is what is called the lysosome. So we've seen this word lyse before, I think. Maybe not in this class, sorry. Maybe we haven't seen this. This means to break apart. So lysis means to break apart, and some just means body. And so this is an organelle that contains enzymes. For what we are going to call intracellular digestion. So this is inside the cell. So if you think about single-celled creatures like amoeba, they have to have some way of getting energy in and nutrients into their body. And so they just simply surround the food and they take it into a vacuole, a food vacuole, and then they digest it using the lysosomes. And so the lysosomes are really important in this function. And so there is a diagram in your book that shows how a cell can feed. So this is a food particle, right? This is my lysosome, which contains enzymes. It fuses with the food vacuole, and then we get it breaking down, and it could possibly use the food for that. So one example of this is phagocytosis and white blood cells. So unlike red blood cells, white blood cells have an immune function. And they can go and they can scout the body looking for foreign cells, and foreign cells perhaps like bacteria. And so phage means to eat. Cytosis means the cell, right? So this would be like white blood cells eating bacteria. So they can go through the body and they can find the bacteria and then they can just digest them. And they can use the nutrients for their own growth and reproduction possibly, and so for their own energy. So I have a live video action showing a neutrophil, and the neutrophil, this is a real short video, but the neutrophil is hunting down a bacteria. So in this image, this is the white blood cell. These are the red blood cells. This is the bacteria. So notice how much larger the eukaryotic cell is than the prokaryotic cell, which would be the bacteria. So the neutrophils are able to detect the bacteria because the bacteria is producing a chemical. So it's going to look like it's actually moving, chasing the bacteria, but it's actually following the chemical trail that is laid down as the bacteria moves. And then you're going to see it engulf the bacteria, take it to the inside where intracellular digestion will take place. There's more than one bacteria there. Okay, so that right there was phagocytosis. And so now it's just moving on, right? It's taken in, it's engulfed it, and it's going to digest it. Okay. Okay, there is another type of digestive organelle, which is called a peroxisome. And you might be familiar with this idea, peroxide, because we can talk about what is called hydrogen peroxide. Hydrogen peroxide is named because there is one hydrogen for every oxygen molecule. So just think hydrogen peroxide is H2O2. This is the same thing that you buy in order to clean your wounds. So you probably have a bottle of hydrogen peroxide in your house. It's diluted extremely, right? So instead of H2O, it's H2O2. So there's one hydrogen for every oxygen atom in the molecule. So this is going to break down into oxygen plus water. Okay. So if you pour hydrogen peroxide into a wound, what does it do? It fizzes, it bubbles, right? And the reason why it's bubbling is that once it's exposed to air, oxygen is produced. Okay? So that is aerobic. So there's lots of oxygen being produced, and oxygen breaks stuff down, right? So specifically, when you use it in your wounds, right, oxygen is, what I want to say, very reactive. And it can kill anaerobic bacteria. So hydrogen peroxide is really good to use if you get a puncture wounds, because the wound is very deep and there's probably not a lot of oxygen there. And so bacteria that survive in an anaerobic environment are going to be present and they're going to start to reproduce. But once you put hydrogen peroxide on it and it fizzes, oxygen gets inside the wound and it kills the anaerobic bacteria. So peroxisomes use hydrogen peroxide. And one of the ways that they also function is that they break down lipids. Specifically. So fats, they break down the fats. And so that occurs in the peroxisomes rather than in the lysosomes. And we'll talk about some diseases, genetic diseases, that are due to a buildup of lipids in the body. And one of those is Tay-Sachs. So Tay-Sachs is a genetic disease that is common in certain populations that is due to defective enzymes in the peroxisome. So defective peroxisomes. Okay. So the other thing I want to talk about are vesicles. Oops, there's only one S. Always want to put two S's there. Vesicles. So vesicles are membrane-bound areas used in transport. And the transportation of the vesicles throughout the cell is regulated by a couple of things. So we have what is called a cytoskeleton. And the cytoskeleton is protein. So the skeleton of the cell, there's protein in the cell, and it gives it its size and its shape, and it also determines how things move, okay? So this is important in maintaining shape, allowing cell movement, and when we get to cell reproduction, it actually pulls the chromosomes apart. So the cytoskeleton is really important. But it also is used internally as transport. So it's like a railroad tracks. So it determines how vesicles are going to move. So it's how vesicles move. Okay. So you see a vesicle being produced at the Golgi apparatus, and you're like, well, how does it know to move towards the outside? And the reason is that it actually, there's a little railroad track that aids in its transport. Also involved in the transport of the cytoskeleton are what are referred to as motor molecules. And these were in the news last year because there was a Nobel Prize awarded to the chemists that figured that the motor molecules actually, if the cytoskeleton is the railroad track, they would be like the train. So they attach to the vesicles and they carry the vesicles. So they carry vesicles. And they're really cool because they're like little machines that are super tiny and that are just composed of atoms, right? They're just a combination of atoms, and they're able to move. Okay? So if we look at a simulation of what this would look like, this is one of my, I love this video. There's a longer version. This is the short version. So what this is, and I'll turn on the sound. I think that music playing with this one. Okay. So what this is a neuron. And in the neuron, we produce lots of neurotransmitters, and they're going to be secreted to the outside. But in the neuron, the neurotransmitters are produced in the cell body, and then they have to be transported to the very end of the neuron, which could be a long distance, right? And so this would be the cytoskeleton down here. This is my motor molecule, and then this is my vesicle. And so they've discovered that this is how vesicles know how to move. And if you look at this motor molecule, you can see the individual atoms, right, that make up the motor molecule. So if it moves, okay, so that is the move. That takes energy to do that because that's mechanical movement. But that is what is going on inside of your cells that allows the vesicles to be transported to the outside of the membrane and then to be secreted to the outside. I don't know, I can't remember if this clip actually has a finishing. Move it along. I think it just goes to the very end there. Okay. So interestingly, chemists are trying to come up with new molecular machines. So can we synthetically produce molecular machines? And so here's an example of a molecular machine that was used or that was produced and that was done by the people that won the Nobel Prize. So see that motor, right? It's like a little, it can move, right? So it's a molecule that is able to move. Okay. So you might hear about nanites in all the recent science fiction, right? Or nanobots, right? That will go into our body, that we put into our body, and they'll be able to go places and then repair the damage, right? So you see that a lot in science fiction these days. So it might be the beginning of it, creating those little motor molecules that can move around in our body. Okay, any questions about that idea? Okay, so vesicles can move via the cytoskeleton and the motor molecules moving them along. Okay, so we're going to move to the very last part of chapter 2, which talks about the plasma membrane. And so does anybody remember what type of lipid has a head and a tail? What type of lipid? Triglyceride, steroid, phospholipid, or what? Phospholipids, right? So the plasma membrane is a phospholipid bilayer. Now, when people first started trying to figure out what the membrane was, they noticed that there's lots of proteins associated with the membrane, and they didn't know how the proteins were organized until somebody won the Nobel Prize for this in the 1960s, where when we look at the phospholipid bilayer, we see that there are proteins floating around in it. So I'll put with floating proteins. And this is what is called the fluid mosaic model for the plasma membrane. So the fluid is that things float around in the plasma membrane. They're not necessarily static. Sometimes they can be static, but generally they float around. And the mosaic model means that different proteins are floating around. So if we look at a diagram of this model, this is what we think our membranes, our plasma membranes look like, okay? The reason why it's called a bilayer is that there's 2 layers of phospholipids. Remember that the heads are hydrophilic, so they're attracted to the water both internally and externally. And then the tails are hydrophobic because they are nonpolar. So they are repelled by water, so that it arranges itself like this. You'll also notice that there are proteins embedded. These can float around and move around inside the membrane. And then there's also cholesterol. So cholesterol plays a kind of an interesting function in the membrane. The cholesterol helps to keep the membrane fluid under cold temperatures. So as the temperatures decrease, our membrane becomes less fluid. And so the cholesterol is really important. And so they find that animals that hibernate, specifically mammals, or if you think about fish, fish are generally the same temperature as the water surrounding them. So imagine what they're like in, the Umatilla River in the middle of winter, right? They would be really cold. So they find that the cholesterol helps to keep the membranes fluid under cold temperatures. So I'm going to write that down. So we have phospholipids, we have proteins floating around, and then we have cholesterol, and this keeps the membrane fluid. Another reason why you have to have cholesterol in your diet. Keeps the membrane fluid. Cold temperatures. Okay. Okay, so this membrane is semi-permeable. Okay, so we're going to talk about how things move across the membrane, because obviously things have to come into the cell, things have to go out, right? And you have to regulate that internal homeostasis, the internal environment. So we say that the membrane is permeable to. Okay. So that means what can move directly across the phospholipid bilayer? What can move directly across without any hindrance at all? One thing that it's directly permeable to is water. So water can move right across the plasma membrane. The other thing that can move would be lipids. We talked about putting steroid cream on your skin and it being directly absorbed. Vitamin A and E directly absorbed. So lipids, and then we have oxygen and carbon dioxide. And so oxygen and carbon dioxide can move across the surface of the lining of our lungs to get into our blood. So that it has to be permeable to that. Okay. It is impermeable, which means not permeable. It's impermeable to large organic molecules. So, for example, proteins. So proteins generally get stuck inside unless they're somehow transported to the outside, secreted. Like enzymes would be stuck inside unless they're secreted. The other thing that they're impermeable to is charged particles. So ions. So sodium and chloride. So sodium and chloride generally do not move across the membrane unless they are transported, unless they move through a channel. So they can't go across the phospholipid body. Okay. So the most easiest way that something moves is what is called simple diffusion. And simple diffusion is due to physics It occurs not necessarily in living systems. It can occur outside of the living systems. So for example, if you had a beaker of water and you put 1 drop of dye in it, the dye will diffuse out until it is equally distributed in the beaker of water. So simple diffusion occurs outside of living things, but it also occurs inside of our body. And so this is where substances move from a higher to a lower concentration. So this diagram shows that. So if we were looking at the concentration of these molecules, right, we can see that they're more concentrated here, and so they would simply diffuse across the surface until they reached an equilibrium, until they were equally distributed on one side or the other. Now, one thing that moves by simple diffusion is water. So this could be a water molecule, H2O. But the movement of water by simple diffusion has a specific name. Does anybody know what it's called? Osmosis. Okay. So osmosis specifically deals with the movement of water. So osmosis is something that will just occur, and sometimes our bodies have to act against osmosis. Sometimes our body uses osmosis to move water, right? So we could get rid of water in our urine, and the kidney uses osmosis to do that. It uses osmosis to filter the blood and get rid of the stuff that's water soluble. So we're going to look at osmosis in lab today. So I'm going to stop there, and then we will finish our discussion of how things move across the membrane on Wednesday.